Hello everyone, my name is Lucian Ilie and I'm going to talk about E-score, which is a new scoring procedure for aligning proteins that we are going to prove is better than Blossom. A few words about protein sequence alignment. We have this fundamental concept in biology that sequence determines structure, determines function, which means all information we need is in the sequence. Similar sequences, significant similarity is not random, is not by chance. So significant similarity implies that the sequences have similar properties. How to look for similarity? We have to compare sequences, and this is done by using sequence alignment. This is arguably one of the most used procedures certainly in biological sciences, but even in all of sciences. The two BLAST papers have almost 200,000 citations, and the use goes way beyond that. When we identify such similarities, these are multiple sequences aligned here. These are usually functional domains. To compute protein sequence alignments, we use algorithms, exact ones such as dynamic programming, which is shown on the right here, or heuristic ones such as BLAST, and they all optimize or maximize some score. Therefore, the scoring matrix is a key ingredient here. The Blossom matrix has been the standard for 30 years now. The problem it is fixed. Two amino acid residues will have the same score independent of the context they appear in. We would like to have, and we will propose, a contextual scoring scheme. The idea comes from natural language processing, where embeddings, which are vectors associated with words, can be produced by training various architectures on very, very large, unlabeled textual data, such as the Common Crawl, Wikipedia, and so on. What's very interesting is that co-occurrence patterns can capture meaning, as shown here on the right. If you take the vectors for king minus the vector for man plus vector for woman, you get something very close to the vector for queen. Meaning depends on context. Here is an example where the word bank has 4 different meanings. And contextual embeddings were the next generation of vectors associated with words. And here are examples, the famous BERT or T5. They help solving difficult problems in natural language processing, and the best ones use the transformer architecture. The idea can be used for proteins, where amino acid residues are used as words and protein sequences as sentences. We can obtain various protein embedding procedures, and for any given one, denoted by E, we define our new scoring scheme. So the E score between two amino acid residues is the cosine similarity of their embedding vectors. So the cosine goes from maximum value when the vectors are similar to minimum value when vectors are opposite, and it's 0 for independent vectors. It is just to have an idea what these matrices look like. So this is the Blossom 45 matrix, and these are averages because these are contextual, so it's not a fixed matrix, but these are some averages computed for various embedding methods. And the Pro-T5 is actually the most similar with Blossom 45. If we compute correlations, we will see that the Pro-T5 is actually the closest to the Blossom matrices. We are comparing Pro-T5 score with Blossom 45, which we proved to be the best Blossom for our experiment, and this is the best E-score. We need alignments, reference alignments, which are not algorithmically but biologically computed. So we picked many varied ones from conserved domain database. We pick 1000 pair randomly from each MSA. We compute using dynamic programming 2 alignments, one using Blossom, one using Pro-T5 score, and use alignment distances to measure how close those are to the reference. The average distance is reported in this table. And as you can see here, except the first case, which is the shortest alignment, these are sorted by length, except the first one, Pro-T5 is better. This is averages, but we can look at the number of tests where Pro-T5 is better, and this is plotted here where this is increasing alignment length, and we see that for the longer alignments, which are also the most difficult ones, Pro-T5 score is better than Blossom in almost all test cases. Actually 94%. Blossom is 4% and 2% is a tie. Most remarkably, even the weakest e-scoring methods still beats Blossom clearly. So we hope this is going to replace the third-tier standard that was provided by the Blossom matrices. Details are found in our paper, which was published here. Thank you.